Welcome back to the class, children. In today's class, we are going to discuss about two concepts. The first one is reciprocal crosses. The second one is back crosses. Let us enter into the concept. Reciprocal crosses. Let us see the definition. If a cross is performed by reversing the sex of the individuals, such crosses are called as reciprocal crosses. Right? Reciprocal means reverse. Right? We are inversing. Right? So let us see an example to get a much more better understanding. Right? So imagine there are two groups. There is a group called as group A, likewise another group called as group B. So we have two groups, group A and group B. So group A has male and female individuals. Similarly, group B also has male and female individuals. So both the groups, both are separate groups. Both the groups have male and female individuals. Now the crosses are performed. So in first cross 1, from the group A, males are selected and they are allowed to mate with the females of group B. So this is cross 1. In cross 1, group A, males are selected and crossed with females of group B. Whereas in the second cross, from the group A, females are selected and they are crossed with the males of group B. So look at the definition or look at the image. So in the first cross, males are selected from group A, whereas in the second cross, females are selected from the group A. So the sexes have been changed or reversed. So this is what we call it as reciprocal cross. இந்த தமிழில் சொல்லும்பொழுது பொண்ணு கொடுத்து பொண்ணு எடுக்கிறது அப்படின்னு சொல்லுவாங்க இந்த கல்யாண வீட்டில். அந்த ஸ்டேட்மெண்ட்டுக்கு நமக்கு அர்த்தம் புரிஞ்சிடுச்சுன்னா reciprocal cross can be easily understood. Right? So when the sexes of the individuals are reversed while performing the crosses, then such crosses are called as reciprocal crosses. So what is the goal of the reciprocal cross? Why we have to perform the reciprocal cross? So this reciprocal cross is performed to prove whether any trait, whatever the character, right? So if there is a trait, if that particular trait, whether it is sex dependent or sex independent, right? Whether a particular character or any sex, the male or the female sex, has any influence in the inheritance of traits. So to identify that, we are doing the reciprocal cross, right? So let us get a clear explanation by observing this diagram, right? So see, look at this image. So here, this is cross 1. This entire side is cross 1. Towards the left hand side, it is cross 1, and towards the right hand side, it is cross 2. So in cross 1, and we are performing the reciprocal cross. Mendel itself performed the reciprocal cross, right? So in cross 1, this is the tall plant, right? We are performing the reciprocal cross for the stem height, a monohybrid reciprocal cross. So in this cross, the Mendel has selected the tall plant and he crossed the tall plant with dwarf plant. So we know, so the progenies were tall. All the F1 progenies were tall. So in the first cross, what he had done, so he had selected the male part from the tall plant, that is the pollen from the tall plant, and he dusted the pollen of the tall plant to the gynoecium of the female plant. So in the first cross, tall plant is considered as the male or taken as the male, whereas the dwarf plant is taken as the female, right? So here look at this flower image. So here he cut and removed the male parts, that is the pollen, and he dusted the pollen over the gynoecium of the female part, right? So he performed the monohybrid cross. So in this cross one, the tall plant, the pollen of the tall plant is taken and dusted over the gynoecium of the dwarf plant. So what he had obtained is all the F1 progenies were tall. So this is the one which he performed and he got the result as tall plant, right? Whereas now he performed another cross, right? So in this cross he collected the pollen from the dwarf plant, right? Now the dwarf plant is kept as a or considered as a male. So now the pollen grains are collected from the dwarf plant and dusted over the gynoecium of the tall plant. Now the gynoecium is considered as a female, right? So look at this image clearly. In cross 1, tall plant is acting as a male, whereas in cross 2, the tall plant is acting as a female, right? Likewise, in cross 1, the dwarf plant is acting as the female, whereas in cross 2, the dwarf plant is acting as a male. So even though he reversed the sexes, he obtained the F1 progenies as tall. And this proves, and that is how Mendel understood, even though the sexes are changed or reversed, the F1 progenies are not affected and all the F1 progenies were tall plants, right? And thus he proved the sex has no role in the inheritance of traits. This is according to Mendel, and this is how Mendel performed the reciprocal cross. And according to the Mendel's monohybrid reciprocal crosses, the sex has no influence in the determination of characters, right? So, and the objective of reciprocal cross is to identify whether the sex of the parents has any influence on the inheritance of traits, right? So, to identify if there is any character is influenced by the individual's sex, right? That is to identify that the reciprocal crosses were performed, right? The next concept is back cross. So, what is a back cross? The cross between the F1 hybrid with any one of its parental genotype. So this is what the definition which has been given in the book, right? So when the F1 hybrid, right, it may be considered whether the unknown genotype, right? So if an unknown genotype is crossed with the known parental genotype, then it is also considered as a back cross. But in our book, it is given in a simple meaning. So the cross between the F1 hybrid with any one of its parental genotype is called as back cross, depending upon which parent we are crossing the F1, right? Whether the F1 hybrid is crossed with the dominant parent, then it is said to be a dominant back cross. When the F1 hybrid is crossed with the recessive parent, then it is said to be a recessive back cross, which can also be called as a test cross. So back cross means when the F1 is crossed with any of the parental variety, it is said to be as a back cross in general, right? So when it is specifically crossed with dominant parent, we say it as a dominant back cross. When it is crossed with the recessive parent, we call it as a recessive back cross or test cross. So this is the chart to explain the dominant back cross and recessive back cross, right? So this is the dominant back cross, and we know this is the F1 hybrid, monohybrid, right? So we have the heterozygous condition, right? So one thing I have to explain you, which I failed to explain in the last video, right? Monohybrid cross. While explaining about the monohybrid cross, right? See, let us see. See, so here this is the dominant plant and this is the recessive plant. So the dominant plant is tall, right? The recessive plant is dwarf. So the first letter of the tall is T. Since it is the dominant, the capital letter to indicate the dominant nature, we use the capital letter or the uppercase. So it is indicated as two capital T's. Two capital T's, it denotes the two alleles of the genes, right? So two capital T's. Whereas if you see this image, here instead of writing the letter D, it is denoted as small t or lowercase t. Why it is used so? Right? If it is a dwarf plant, it is a recessive plant, the small d has to be written. Two small d's. But instead of writing the D for doctor, right? Instead of writing the D, we are using the lowercase of t, that is the lowercase of the dominant form. So why they are using in such a way, right? In general, to avoid the confusion, right? For a better understanding, whatever the alphabet which is used to denote the dominant nature, the lower case of that alphabet is used for denoting the recessive gene, right? So, if this is a tall plant and if this tall plant is denoted by the capital T, then its recessive form will be denoted by the small t's. Or by example, if this is denoted by the capital letter XX, right? So if it is denoted by, if the dominant is denoted by capital X, then the recessive will be denoted in the small letter x, right? So just to understand in a better way and to avoid getting the confusion, so whatever the alphabet which is given for the dominant, the lower case of the same alphabet will be given to the recessive gene for easy understanding, right? Now let us go back to the dominant backcross. Now this is a F1 hybrid, right, having a heterozygous condition, right, and when this F1 hybrid is crossed with a dominant, homozygous dominant, we know homozygous dominant, both the alleles have same, right, they both are same. So this F1 hybrid produces two different types of gametes, whereas the homozygous dominant produces similar type of gametes, and when they are crossed, right, when they are put in the Punnett square, all the progenies obtained as a result of dominant backcross were tall, right? So always in dominant backcross, right, the progenies has to be of similar phenotype, right? So that is what we have to keep in mind, right? So always the dominant backcross will show the similar phenotype, right? So that is the phenotype will always be dominant, right? Dominant phenotype. So only the dominant phenotype will be expressed in the dominant backcross, right? So all the progenies of dominant backcross will be dominant only. There won't be any recessive trait seen in the dominant backcross. So whether it may be a homozygous dominant or heterozygous dominant, that doesn't matter, right? Always in dominant backcross, the progenies will be a dominant phenotype, right? Whereas in the recessive backcross, which is also called as test cross, the F1 hybrid is crossed with the recessive parent, homozygous recessive parent, F1 hybrid producing two different types of gametes, whereas the recessive parent producing similar type of gametes, and when they are crossed and when they are put in the Punnett square, we get 50% of tall plant and 50% of dwarf plant, right? So here in recessive backcross, always the phenotypes will be obtained in equal proportions. Whatever the phenotype we are obtaining, right? So whatever the phenotypes are there in the plant, right? Whatever the phenotype we are considering, all the phenotypes, if it is a monohybrid cross, we have considered, so tall and dwarf alone, right? Single character is considered, and single character with two contrasting traits, right? So both the characters will occur in both the phenotypes will occur in a equal proportion. So this is the difference between dominant backcross and recessive backcross. In dominant backcross, only the dominant form, 100% the dominant progenies will be obtained, or the dominant phenotype alone will be obtained as a result. Whereas in recessive backcross, all the phenotypes will occur in a equal proportion, right? So here in monohybrid cross, we have tall and dwarf nature. Both the tall and dwarf nature have occurred in the equal proportion, 50-50, right? 1:1. So always in recessive backcross, for a monohybrid recessive backcross, the ratio will be 1:1, right? Because we are considering only one character right? One character has two different forms, the dominant and the recessive, right? So here tall and dwarf in the equal ratio 1:1, right? So the progenies of the recessive backcross will show both the dominant and the recessive triads in equal proportion. So this we have to keep in mind. So this is the difference between the dominant backcross and recessive backcross, right? So objective of test cross, right? Test cross is nothing but the recessive backcross. So test cross is usually performed to identify whether the genotype of an individual is homozygous or heterozygous, right? So if see this image, so if this hybrid, if only if this hybrid is a heterozygous, the ratio will be 1:1, right? So you do the crosses, you can able to check it out, right? You can able to check it out and find out how it comes, right? So only if the F1 hybrid is a heterozygous, we can able to obtain the ratio as said, that is 50-50, right? Or 1:1. If this F1 hybrid, if it is a homozygous, for example, imagine this is capital T, both are homozygous dominant, right? So then the ratio won't be 1:1 or 50-50 ratio cannot be obtained. All the plants will be dominant. Or if it is a recessive, both are small t's, right? If it is a recessive plant, then at that time all the plants is going to be a recessive, right? Until and unless if this is a heterozygous, we can able to get the ratio as 1:1, right? So thus, if we don't know this, what is the genotype of this F1, right? To identify what is the genotype of the F1 hybrid, if this F1 hybrid, imagine this F1 hybrid genotype we doesn't know, right? If this is crossed with the recessive parent, because recessive parent we know the genotype. When this unknown F1 hybrid is crossed with the recessive known genotype, we can able to get the result, and if the result is in equal proportion, then the F1 hybrid is a heterozygous. If the result is either dominant or recessive, then F1 hybrid is a dominant or recessive plant, right? So this is the main objective of the test cross, right? Test cross is used to identify whether the genotype of an individual is heterozygous or homozygous. If the result of the test cross is same, right, either dominant or recessive, then it is said to be a homozygous hybrid. If it is said to be a homozygous plant. Whereas if it occurs in the equal ratio 1:1, then the F1 hybrid is said to be a heterozygous, right? Yes, children, hope today's class will be a useful one. Let us meet you in the next episode. Thank you.